the English language. And after all the presentations are complete, we will have a 20-minute section for question and answers from you, the audience. So I ask that put your questions on the chat box, and so I'll be able to read them all and to be able to read them to the speakers so they can be able to answer to all your questions. We have 3 presentations today, and I really hope that you all enjoy this event. This event today is about share the experiences gained from the validation of COVID-19 diagnostic tests and how this can be applied to the validation of diagnostic tests used in seed health diagnosis, because validation is a requirement of method development, and it is a process that determines the fitness of a method for its intended purposes. The International Seed Health Initiative for Vegetable Crops, ISHI detection methods are used for many purposes, and such as preventing pathogens moving from one country to another as a pre-screen to clear lots that are healthy for commercialization and for seed producers to make management decisions. Method validation is a critical aspect of method development in ISHI, and performance criteria such as analytical specificity, analytical sensitivity, selectivity, repeatability, and reproducibility are measured to demonstrate that the methods are fit for purpose. We would like to start with our first presenters, where they will be talking about performance validation for SARS-CoV-2 in vitro diagnostic tests. And to be able to present this, I would like to welcome Dr. Irena Prat and Dr. Ute Ströher. Dr. Irena Prat holds a degree in laboratory biomedicine and a postgraduate degree in management. And prior to joining the World Health Organization, she served as a medical devices counselor at the Agency for Medicine Products. And Dr. Ute Ströher is currently a technical officer in the pre-qualification team for in vitro diagnostics within the Division of Access to Medicines and Health Products at the World Health Organization. Her responsibilities cover the emergency use listing procedure, as well as development of technical guidance and a specification series documents. And now I give you the floor to Dr. Irena Prat and Dr. Ute Ströher from the World Health Organization. The floor is all yours.
Thank you, Irene. Can you confirm if I'm in presentation mode?
Not yet.
Not yet.
Yes, perfect.
Okay, excellent.
Thank you very much, Chair, for the introduction, and hello, dear colleagues. It's a pleasure to be present at this event. I'll share this presentation with my colleague Ute, who will go more into the technical details. But before we start looking together at some validation rules and responsibilities for this type of product, I would like to mention that our team sits in the pre-qualification unit at WHO. And our main task in normal times, I would say outside of pandemic, is to run what we call a pre-qualification program for in vitro diagnostics. This program was established by WHO to assess in vitro diagnostics for their safety, quality, and performance characteristics. And the focus of this program is on priority diseases and their suitability of specific in vitro diagnostics for use in resource-limited settings. You can see on this slide the areas of work in which we normally focus, but since the pandemic has been in place, obviously we have shifted to other activities you will hear about in just 2 or 3 minutes from now. Next slide. Next slide. So the findings of the pre-qualification program are used in 2 different ways. First of all, by WHO member states, when they are trying to select from a wide variety of products which are actually on the market, and to be able to know which ones are of acceptable quality, safety, and performance, they look at the list of WHO pre-qualified products. The same list is also used by international donor agencies that donate in vitro diagnostics to countries which are in need. And obviously pre-qualification listing, which is a brand of quality, is used by both member states and also donor agencies in conjunction with other procurement criteria, which are equally important when selecting products. Next slide. So pre-qualification will look at each individual product, each individual in vitro diagnostic through a procedure which is standardized and applied every time in a consistent manner. And this procedure and process actually include three key components. The review of a product dossier, which is compiled by the manufacturer by selecting specific records from their technical file, a performance evaluation, which we coordinate in collaboration with one of the laboratories that work with us, and which is an independent verification of some key performance and operational characteristics, and a manufacturing site inspection, which complements the review of safety and performance, which is done through the dossier review and performance evaluation by auditing the manufacturer for the quality management system. So the combination of these three activities, in addition to the labeling review, which is done across the three activities, allows us to get a comprehensive insight into product characteristics spanning across safety, performance, and quality. Next slide. So now looking at what has been actually our key activity for the last year and a half, which is called the emergency use listing procedure. This procedure was developed back in 2014 when we were facing the Ebola outbreak. And it's intended to assist, again, interested procurement and donor agencies, as well as member states, on the suitability for use of specific in vitro diagnostics, but which is a difference to pre-qualification based on a minimum set of available quality, safety, and performance data. So this is a risk-based approach, aiming at expediting the availability of in vitro diagnostics which are needed in a public health emergency situation. The information we request from manufacturers in this kind of context is different from the one and is different from the procedure we use under pre-qualification. So we will still be looking at the quality management system review, but based on a more limited data set. In this case, we will perform a desktop audit of key quality management system documents, and we will not go on a site inspection. And the second data set, which is equally important, is a product dossier review, where we will assess the documentary evidence of safety and performance of the product. Next slide. And over to you, Ute.
Yeah, thank you, Irena. So this slide just gives you one overview of the key differences between the pre-qualification and the emergency listing. And as Irena pointed out, the three pillars, and I won't go into detail now again here. And in the rest of the talk, I will focus on the technical documentation and the performance assessment. So similar to what we do for pre-qualification, we develop specific requirements to manufacturers outlining the validation criteria for specific diagnostic tests. And we have done that also for SARS-CoV-2 diagnostic tests. So we have two documents. One is summarizing, not really summarizing, detailing the specifications for molecular tests, so for the detection of nucleic acid of SARS-CoV-2 and antigen detection, and the second document for the detection of antibodies. And both documents are updated based on new evidence that become available, and both documents are available on our website. So in these documents, we clearly describe what manufacturers need to submit in order to obtain emergency use listing. So they have to provide a detailed product description and what kind of regulatory like regulatory versions of the product exist and which one they're applying with. It needs to contain a risk analysis, detailing the different risk to patients or community, etc. We expect detailed product design information, not only the formulation of buffers also, but also the sequence information of primer and probes in order to verify, for example, any in silico analysis claim, etc. The product performance specification, they will follow on the next slide. And as Irena mentioned, we're also doing a labeling review. And this clearly looks on the instructions for use. So does the claim the manufacturer makes is supported by the validation data they submit? And also are these claims are in line with existing testing recommendations by WHO? So for the product performance specification, we basically ask the same information. We ask for product that are submitted for pre-qualification. However, we call them light because we don't go, or we don't require as much in-depth data. But and I'm going to briefly through them. So we expect manufacturers to demonstrate specimen stability, informing the user on how to collect the specimen, how they are processed, and then how long the specimens, for example, are stable in extraction buffer or unextracted, etc. Then there is a validation of specimens to basically demonstrate equivalency for PCR tests. That is, for example, if a manufacturer would claim oropharyngeal swab and saliva, that is going, that the saliva would have any inhibitory components that was impact the performance. So this is really looking for matrix inhibition and less for clinical equivalence. Then, of course, we ask for precision studies, analytical sensitivity, the stability of the product, analytical specificity, so with the aspects of interference and cross-reactivity. As I mentioned before, the prime and probe choices need to be justified and made available. For antigen tests and antibody tests, there needs to be evidence, or the high dose hook effect needs to be investigated. And procedural controls also need to be validated. What I put here in bold and larger is we are asking for flex and robustness studies, and this is taking into consideration that we are mainly focusing on low and middle income countries, and that might mean that the operational temperature is not the same like in a well air-conditioned labs in the US or Europe, and several environmental or operational circumstances that might be different in the low and middle income countries than in high income countries. And then, of course, the clinical evidence, where we're asking for clinical sensitivity and specificity. And here, I think it's quite clear that we are not asking in regard to number of specimens as well of geographic region. We request lower numbers and less geographic region that we would ask for product that we want to pre-qualify. And I listed just here some factors that actually consider not only the R&D and the risk analysis for the manufacturers, but also the validation, and that impacts the requirements we drafted. So obviously the risk class of the IVD that is determined by if a false a false result, the impact on individual health risks and the public health risk. So the highest risk class is a category D, and that, of course, undergoes the highest scrutiny and assessment and validation. Then it's important the specimen itself. Which kind of specimens are actually recommended? And for SARS-CoV-2, at the beginning, it was mainly lower respiratory specimen like sputum and oral and nasopharyngeal swabs. By time, there was also some evidence for nasal swabs, but this involves. So there really needs to be focus which specimen types are suitable, and there needs to be a balance between non-invasive. So, for example, a saliva specimen is obviously much easier to collect than a nasopharyngeal swab. However, the sensitivity is lower. Then the patient status needs to be taken into consideration. Very critical in the intended use. So is the test meant for screening or diagnostic purposes, for monitoring over time, or does the test identifies or if a treatment can be given or not? So this is very, very important and determines the clinical data and manufacturers need to generate. Then obviously the user profile of the intended user. So for a near point of care or point of care device, the validation needs to take this into consideration. And then in interest of time, just going for the PCRs, in the very beginning, we were accepting single target test, PCR test for SARS-CoV-2. However, with time and the emergence of variant, the requirement now is for manufacturers to submit dual target tests. A big issue are the design of internal controls. So the two common ones are the endogenous controls, that is, there are a primer set that detects a housekeeping gene, and versus exogenous controls, that is, for example, a RNA bacteriophage that you spike into your specimen. Both controls have different different information. The choice the manufacturers made, it needs to be also justified. And there are, of course, pros and cons for each control. And I thought I'm just going for a few issues and challenges we have come across by assessing dossiers. Of course, it's at the beginning the lack of reference materials. So manufacturers used all kinds of things, DNA, plasmid, inactivated virus, etc. So it was very difficult to compare results. The good news is there are now WHO international standards for RNA and antibodies, and one for antigens in development. Then we had issue that the validation studies didn't utilize the entire testing system, so including the specimen preparation and the nucleic acid extraction, which is of course critical and really might change the result. We need to have predefined acceptance criteria by the manufacturers to see what is acceptable to them or not. Again, as I said, the rich nucleic extraction kits and platforms were considered. In regard to stability, it was that we request real-time stability data, but they didn't support the shelf life that is mentioned in the instructions for use, and basically a non-understanding of manufacturers the importance of flex and robustness studies considering low and middle income countries. In regard to the clinical studies, at the very beginning, where clinical specimens were not everywhere available, we accepted a portion of the clinical study to be performed by using contrived specimen, but that was only at the very beginning with a commitment of the manufacturers to support real clinical data later. This obviously is not applicable anymore because clinical specimens are unfortunately available everywhere. Also that claims were made by the manufacturers for several specimen types. Also, there was no clinical evidence to support that. And this is especially critical for if a manufacturer claims the use, for example, of saliva, but doesn't provide the data. And it is known that saliva is less sensitive than, for example, nasal or nasopharyngeal swabs. Then there the clinical study design using retrospectives versus prospective specimens, how how selection bias was avoided, inclusion criteria. So we really require a good explanation if a manufacturer uses retrospective specimens, and we allow only a certain percentage of the clinical study to be performed on retrospective specimens. And then, of course, the intended use claims. Most of the testing recommendations are for symptomatic patients. So if a manufacturer suddenly has a screening claim for non-symptomatic individuals, that needs to be supported by clinical evidence. And this kind of studies are obviously much more challenging to perform by the manufacturers, but it's a requirement. And then data integrity concerns that from case to case are present and are a challenge to address. So once, briefly, I think I'm running out of time, but briefly, so once we list a product, we expect the manufacturers to submit changes they made to the product, and that might be the design or just instructions for use or the manufacturing side. So we need to have this information submitted. And also we expect from the manufacturer that they have a post-market surveillance system in place and are monitoring and informing us about complaints and they receive about the products. And that was, of course, also of high importance when there are some sort of false results due to the emergence of variants. And I'm end here. Thank you very much.
Thank you so much, Irena and Ute, for this fantastic presentation, and of course to provide us with this overview on performance validation inside the World Health Organization for SARS-CoV-2. So this is really good. Thank you so much. And now I would like to proceed with our second speaker of this event today. And we have Dr. Loh from the National University Hospital of Singapore. He will talk to us about the impact of disease prevalence in assessing the performance of diagnostic tests. Dr. Loh was born in Penang, Malaysia, and he currently serves as a consultant chemical pathologist, research director, and director of informatics at the Department of Laboratory Medicine, National University Hospital in Singapore. Dr. Loh, the floor is all yours.
Thank you very much for the very kind introduction. Hi, everyone, I'm Tze Ping, and today I'm going to talk to you about the impact of disease prevalence in assessing the performance of diagnostic tests. I have a few acknowledgements. First of all, I would again like to thank the organizers, the International Seed Federation, for the kind invitation for this really unique opportunity to cross-pollinate with our colleagues. And I have no conflict of interest to declare for this lecture. And most of the information or details of the lecture that I'm delivering now can be found in the following publication. So for interested audience, you may refer to this paper. So when COVID-19 outbreak started, WHO very quickly sort of gave the recommendation that the key strategy to diagnose or contain this outbreak is to perform test. We need to identify patients. We need to test at-risk subjects, and we need to have the appropriate treatment and quarantine strategy for these subjects that we have identified. So never before in laboratory sort of history that we have such strong emphasis, not only by the government agency or the healthcare agency, but the awareness of the public about the importance of testing, as well as their appreciation of different aspects of testing. So it's rare indeed to have lay press discussing as well as explaining the importance of test sensitivity, test specificity, sometimes even going into predictive values to educate the public about what does a positive result means for them. So laboratory tests for SARS-CoV-2 or the virus causing COVID-19 have several main modalities, including laboratory-based antibody tests, polymerase chain reaction tests, PCR, and rapid antigen tests. For diagnosing an acute infection, PCR is preferred. And in terms of the availability of SARS-CoV-2, as of 26th of August 2021, we have about at least 300 commercial laboratory assays that have received FDA approval and are listed on their websites. And as we heard from the preceding lecture, these involve very detailed consideration of various aspects of the assays, including the analytical performance, but also the robustness, as well as taking into account how they may perform in field conditions. Having said that, no assays are perfect. So all assays would have its own false positive rate as well as false negative rate. And that in turn defines the sensitivity and specificity. So the assays have differing claims of clinical performance, such as clinical sensitivity and clinical specificity. So this is just an example of the listing of the available COVID-19 tests. And as we can see from the table, the sensitivity can range from 85% all the way up to 100%, and the specificity can range from 92% to 100%. So how does one select the appropriate assay? So we heard again from the preceding lectures of how they go about regulating as well as approving the assays. But when it comes to individual laboratories, we have to make the decision of selecting one out of the 300 that are available to us. So the primary consideration must be that the assay can fulfill the intended clinical purpose of the laboratory test. And when we decide on the intended clinical purpose, we have to consider the clinical consequences of false results. For example, a false positive result would expose the virus to the individual, whereas a false negative result, where a patient is positive for COVID, but then tested as negative on the test, and therefore released back into the community, may expose the community to the virus. So therefore, it's important for individual laboratories to have a priori defined predictive values criteria that can then be applied to objectively select assay that fits the purpose. So predictive value is basically the probability of an actual underlying status based on disease prevalence. For example, a 90% positive predictive value means that if a test is positive for a subject, there is a 90% probability that the person has the infection. So this is, I'm just going to show a few graphical representation. So in the low prevalence setting, assuming an assay has 90% sensitivity and 99% specificity, and therefore we have 10% false negative and 1%, 10% false negative and 1% false positive. A positive result has about 50% probability of having a disease, whereas negative result has 99.9% probability of not having a disease. So this is because we have a vast majority of the subjects being healthy, and therefore even at a very, very low false positive rate, when we have one subject with a positive result, it would lead to about a 50-50 chance that a positive result has a positive disease. By contrast, in the same low prevalence setting, if the sensitivity is very, very high, of 99%, and therefore with 1% of false negative, whereas the specificity has dropped to 90%, meaning we have 10% false positive, then the positive result is only associated with a 10% probability of having disease, whereas a negative result has 99.9% probability of not having disease. So next, we examine a situation where the prevalence has increased from 1% to 10%. And again, we have two sets of assays. One is with 99% sensitivity and 99% specificity. And again, we can see that with increased prevalence, the positive results, the positive predictive value has actually improved to 91%, whereas the negative predictive value has marginally decreased. Conversely, if we have a moderate prevalence, but we switch over the sensitivity and specificity such that we have now 99% sensitivity and 90% specificity, a positive result has about 50% probability of having disease, whereas a negative result has 99.9% probability of not having disease. Put in another way, in a graphical format, what this tries to show is that at relatively low prevalence, negative predictive values can be easily fulfilled by sensitivity, and this is what we meant by that. So if we have a 1% prevalence, we can see that all the lines that fulfills the various negative predictive values are nearly at zero, meaning that we need very minimal sensitivity to achieve high predictive values at low prevalence setting. And by contrast, in low prevalence setting, we would need high specificity to achieve high positive predictive value. So positive predictive value is much more demanding of specificity. So when we put all these together, the key observations that we can find is that sensitivity and specificity exist in an inverse relationship. And when prevalence is low, the test needs to be more specific to lower false positives. And when prevalence is higher, then the test needs to be more sensitive to lower false negatives. So as a laboratory, we need to define the predictive values working in conjunction with the end users of our laboratory test results. And we should work out what is the desired predictive value for their clinical setting in the prevalence whereby the test is applied in. We then work back to find out what is then the minimum clinical sensitivity and clinical specificity that can achieve the desired predictive values. And we have made available a simple calculator to calculate the minimum clinical sensitivity and minimum clinical specificity based on desired predictive values as well as prevalence that can be found in the supplemental material in the publication mentioned earlier. After we have selected a test, it is important for us to continue to maintain the tests, as alluded to earlier. This is so that we can ensure that the test continues to perform as what we have a priori defined in the real world in a continuous manner. So a lot of the tests that we run provides numerical results. For example, for PCR, we may find CT values, but are often interpreted qualitatively, whether it is positive or negative. And PCR tests can indeed incur systematic shift or drift due to reagent changes, reformulation, degradation, differences in lot-to-lot reagent preparation, and instrument errors, etc., etc. So when we have a PCR test, we can obtain CT values for the healthy individuals as well as the diseased individual. And so, oftentimes, there are some degrees of overlap. And this is where we draw our threshold to try to optimize the best cutoff points for diagnosing the patients. Graphically, this is represented by the ROC curve, or the receiver operator curve. And we have the cutoff defined on the graph. And when we apply the desired positive predictive value and negative predictive value on the ROC curve, we can actually define the boundary within which the assay must perform in order to continue to fulfill the previously defined positive and negative predictive values. And when we encounter a systematic shift, it is relative to the cutoff values, and therefore we can assume that it is the equivalent of the cutoff points being shifted towards the right or the left. And the distance that the cutoff values can travel is the maximum positive value, maximum positive bias or maximum negative bias respectively on the graph. And therefore, we can then define objectively what is the maximum tolerance in terms of systematic bias or systematic drift or shift for that particular assay if we want to maintain it within this positive and negative predictive value. And these are some examples that we have worked out. In this case, it's not a PCR test, it's an immunoglobulin test, but it nevertheless shares the same underlying principle whereby it reports numerical results, but are interpreted qualitatively. And what we found is that for the most common commercial assays that we have, they do all indeed show differing tolerance for the same positive or negative predictive values that we have determined. And some assays are not able to achieve certain positive or negative predictive value, even at baseline without any bias. And therefore, this is again another graphical way that we can use to also select for assays that whether it fulfills our desired clinical purpose. So again, to summarize, the key observations is that bias tolerance can be linked to the clinical use case, i.e. the predictive values. Bias tolerance should be tailored to specific assays. And bias specifications may be different for positive and negative bias, although it is far more often for us to define positive and negative bias tolerance in equal magnitude. So in conclusion, when we wish to select an assay, it is important to set an a priori predictive value for the assay based on its consequences. For example, false positive risk is to throw away a good batch of seed. False negative is to risk contamination of the rest of the other seeds. And we need to ascertain the prevalence where the assay is applied, and then we can then work back the minimum sensitivity and specificity. Do bear in mind that prevalence is not necessarily what is quoted for the population. In some instances, we may enrich prevalence, for example, by selecting high-risk subjects to be tested. These could be, for example, based on signs or symptoms. And through this enrichment process, we may be able to increase the prevalence of the population that we test. And therefore, for some assays that we were not able to achieve the desired predictive values based on the sensitivity and specificity, we may be able to achieve it after enrichment. And finally, use selected predictive values at given prevalence to inform bias tolerance with receiver operator curve and monitor the performance of assay to ensure that it continues to fulfill a priori defined predictive values. Thank you very much. The flowers shown are orchid, which is the national flower of Singapore. Thank you.
Thank you so much, Dr. Loh. It was a fantastic presentation and very well pointed. Some of the key things that you mentioned about the predictive values, about prevalence, sensitivity, and specificity. So that is incredibly important for us. So thank you so much for this presentation. And thank you for showing the beautiful orchid flower, which is a symbol of Singapore. Now I would proceed with our third and final speaker for the event today. And he is Dr. Harrie Koenraadt from Naktuinbouw, the Netherlands, and he will talk to us about the detection of tomato brown rugose fruit virus and the bridging experience from COVID and seed health tests. Harrie is currently a senior researcher in the R&D detection of Naktuinbouw and responsible for the development and validation of vegetable seed health tests. He started working for seed company in 1992 because of the pepper mild mottle virus outbreaks in Spain. Harrie, the floor is all yours.
Okay, so do you see the presentation right now?
Yes, we do.
Okay, I'll put it in the presentation mode.
Perfect.
So, yep, okay. Well, thank you very much for the invitation. And I would like to talk a little bit about Tobamal viruses. So it's not a new problem. Actually, in 1886, there was already tobacco mosaic virus in the Netherlands, and that gave lots of problems. In 1992, there was pepper mild mottle virus in Spain that gave outbreaks. And actually, at that moment, some seed companies started to disinfect seeds. And we introduced as seed companies the seed treatments and a bioassay to monitor the efficacy of seed treatments. And actually 1992 was the start of the International Seed Health Initiative. And then in 2014, we got a new Tobamal virus on the block, and it was tomato brown rugose fruit virus, which gave a lot of problem. So Tobamal viruses are actually rod-shaped particles, and RNA is well protected by the coat protein. So Tobamal viruses are actually very persistent viruses. So if you have infected pepper, you can eat the sauce or the fruit themselves, and the virus can pass your stomach and your intestinal system, and then still the virus is infectious. So it's very persistent. To be clear, it's a big difference with the COVID virus. Of course, Tobamals are not pathogenic to humans. It's just a plant virus. So also they are very stable. At high temperatures, 80 degrees stable, easily survive. And you need a temperature of 95 degrees for 10 minutes to disinfect virus particles. And pepper mild mottle virus is actually a Tobamal virus that is everywhere, because there are a lot of sources that contain huge amount of pepper mild mottle virus, so they are used as an indicator of fecal contaminations. And everywhere in the world, you find very high concentration from about 1,000 to 1 million copies per liter. So they are very persistent. They're not only persistent, they're also very abundant. You can isolate or purify virus, and then you get a maximum of 10 milligrams per gram of fresh tissue. And so that's a relatively high yield, but 1 to 3 milligrams per gram is more common. And if you you can calculate by that number, and by knowing the molecular weight of 1 mole of the virus, and then multiply that with the number of Avogadro, and you get the number of virus particle per gram. And actually, For Tobama virus, that's huge. It's about 1.5 times 10 to the 13. So we're talking about trillions of virus particles by per gram. There's another way of calculating the number of particles. Actually, Sven Berensen, his lab, actually they made a dilution curve with known quantities of systemic single-strand DNA, and they used a Tobamal virus target to quantify the number of particles in freeze-dried reference material of tobacco mosaic virus. They got a CQ of 8, so you can then use the curve, and that will tell you that there were about 1 times 10 to the 12 variants per ml in that original reference material. So also we are in our lab, we're seeing CQ values as low as 1, 2, 3. So that means actually that you come into the same number that you are talking about, trillion virus particle per gram. So they are all abundant. So actually, there are a way of testing seed lots of pepper and tomato. There is an ELISA that can be used, and if the ELISA result is negative, that means that you have a healthy seed lot. If the ELISA is positive, that means that there is Tobamal virus present on the seed, then you can do a disinfection, and the disinfection efficacy can be evaluated with local lesion assay. If it's negative after the local lesion assay, that means that disinfection was good. If not, then it's an infected seed lot. And so tritium salt phosphate and for instance, hypochlorite can be used to treat seeds of tomato and pepper to reduce the virus load. So actually talking about some of the qualities of the polyclonal antiserum, so the sensitivity is pretty good. The bioassay, it's less sensitive, arguably it's sensitive enough, but for pepper we know it's a bit less sensitive. If you're talking about specificity, it's a polyclonal antiserum, so it cannot distinguish between related Tobama virus, so there is cross-reaction. As for the bioassay, it's the same, but still both the polyclonal ELISA and the bioassay are fit for purpose. The viability cannot be judged with the ELISA, but the bioassay will distinguish between infectious and non-infectious virus. So then in 2014, we saw for the first time TOVRV in Jordan and Israel. The big problem with this new virus is that it's overcoming a resistance, PM2 resistance in tomato, and therefore it's rather damaging. Seed transmission is known, and because it was not present in many places, it got a quarantine status. And with the quarantine status, that gave a problem that the polyclonal ELISA and the bioassay were not good enough anymore to determine whether seed lots were contaminated with TOVRV or not. So we had to work on an alternative method. So TACM actually was developed, and in terms of sensitivity, it has a very high sensitivity and specificity as well. Viability cannot be evaluated. It does not discriminate between infectious and non-infectious virus particles, and cross-contamination that was not known at that moment, but from the ELISA, we knew already that the ELISA is there are some issues with cross-contamination there as well. So probably there are chances of cross-contamination. So how does the TACM works? Basically, a sample is 3,000 seeds is a test in 3 subsamples of 1,000 seeds, and then samples are extracted. Extraction buffer is spiked with an ornamental virus as a process control. RNA purification is done, and then a triplex TACM is performed. There are variations in the protocol between the laboratories in grinding equipment, extraction buffer, RNA purification system. So there are some differences. It's a two-target reaction. So there are two targets, CSP 1325 and CATA 28 are actually two different targets within the TOBV genome. And so the BAC-V is as the process control to monitor inhibition. There is an arbitrary cutoff of 32, and in our lab we use 2000 as a threshold for the signal, but those are arbitrary numbers. So validation, of course, validation is very important to determine whether a test is fit for purpose. So we did that for tomato and pepper seeds, and it's a qualitative method, so present absence. And we want to be sure it only detects BRV, and at least one infected seed should be detected in a subsample of 1,000 seeds. And so the analytical sensitivity was determined using 3 different seed lots, one from Germany and 2 from Jordan, where all seeds were contaminated with TOBV. And basically what we did, we made 8 samples and added 1 of seeds for each seed lot, and then we did we performed the test. What you can see is that for tomato, clearly all subsamples 8 times were found with CQs around 15 average. There is for pepper, it's a bit higher, so we know already for a long time that there is a little bit of inhibition in the pepper matrix. It's actually for both TACMs, you can easily see that one infected seed is detected. But there is a variable load per seed. That's what we know. We don't have nice reference material with an homogeneous amount of virus. There is a matrix effect, so pepper is a bit more difficult than tomato. And the limit of detection, we don't knew at that time the number of copies, which is probably different from the work on COVID, but we know that it's about 1 to 10 copies per PCR. Well, the inclusivity and exclusivity, you want to be sure that you pick up all the TOBV isolates. So the ones that we had in our collection, we tested them and they were all picked up. And the related Tobamal virus are not picked up, indicating that it was specificity was okay. You see that there is a bit of signal generated with, for instance, pepper mottle virus, but that was just above the threshold. So what about the TOBV load in leaves and sepals? We did a study with our NPPO, the NVWA, Naktuinbouw together. We worked with material from a reinfected greenhouse. We sampled different parts of the plant. For instance, the sepals, those are the small green leaves on the tomato, the fruit itself, but also old leaves and unripe fruit, ripe fruit. What you can see when you look into TOBV in symptomatic plants, we actually sampled 5 symptomatic plants and then looked to the variation. What you see is actually that in all the plant parts, you can detect TOBV easily, but old leaves, there was relatively less virus to detect than in younger parts of the plant. So the younger plants, actually, there is much more virus. The top leaf actually is, it has it's very young, so presumably there is most of the virus, but in greenhouses in the Netherlands, sometimes the top leaves are at 4 meters, so you need a ladder to sample. Therefore, there are some nice alternatives, so the sepals are working okay. If you look to the non-symptomatic plants in the same greenhouse, actually, we saw that there was signal in all the different plant parts, which was a bit surprising because we thought, well, there are no symptoms, so they should be negative, what we saw in an earlier experiment. But in this experiment, actually, the older leaves almost contained a bit more virus than the young parts and the fruits, and that was surprising. So actually, if there would be infection, then you would expect a high Tobama load in the ripe and unripe fruits, because those are young parts, and that was actually not the case. So it can be a very late infection, but it's more likely that actually those samples in the same greenhouse, that they have dust on it, and that so they are the Tobama is present on the leaves, but not in the leaves, so it's not an infection. So the sampling of Tobama is quite critical, which is maybe same as for COVID. So what about the Tobama load in leaves and samples? If you look to samples, and you can see that those are about 140 samples that are actually two groups, samples that are with relatively low CQ values and groups with relatively high CQ values. If you put it in a box plot, this is the same information in a box plot. You can compare it with COVID, and so how does that compare? One difference between our work and the medical work is actually that the medical papers, often the low CQ values are down in the graph, and we put them up, so you have to flip it. But for COVID, actually, there are also the virus load is in general lower, but also there are patients with very high CQ10 values of COVID. So also there are high values, but in plants actually there is more two groups, and so the question is whether the groups with very high values, whether that's really biological relevant, or is it what's the meaning of those very high numbers in plants? It's difficult to make a decision on that. So we also look to the Tobama load in seeds, and this is just a 1 month data set, and you can see that there are several samples with values under the 30, so that means clearly that they are positive. They are from one and the same place, so it indicates that it's an infected seed lot. If you look to many more samples, and we also did a ranking of many more samples, you can see that there are seed lots with very low CQ values, about 15 to 20. There are quite a lot of those, and there are also seed lots with relatively high CQ values. And the question is whether that's still biological relevant virus, or that it's just a cross contamination that could occur during the seed processing. So some observations in the Tobama seed and leaf testing program. So there are considerable numbers of samples with a relatively low Tobama load, and that's the question is what is going on with those samples. Are they treated seed? Maybe is then the virus load reduced? Can it be cross-contamination during seed processing, or cross-reaction with related to Tobamal virus? Well, from validation work, we know that cross-reaction with related to Tobamal virus is very unlikely. So then cross-contamination during seed processing is maybe a more logical explanations. There are also a low number of seed lots with high virus loads, and there it's more likely that infection during the seed production, and those seed lots are probably dangerous in terms of spreading the virus. So if we're talking about the seed health assay, the sensitivity is very good, the specificity is very good, the viability we don't know. That's already and chance of cross-contamination is yes. It looks that it's in the lab. You can monitor that by a good laboratory practice and having the appropriate controls. You can see that. So it's more likely that during seed processing that there is some cross-contamination between samples. And for the leaves and the sepals, we have several samples with very high Tobama load. So those are just infected samples from infected greenhouses, and several samples with low to very low virus load. It's more logical that it's dust on leaves on greenhouses that previously were infected. Of course, you can never tell that it's a late infection. So what is happening now is often that there is resampling to be sure that to get a good reading on those greenhouses. So, but it looks that there might be dust on the leaves. What about the genetic variation and the origin of TOBRV? So on the EPO, there is of course a geographical distribution as shown. And you can see that TOBRV is already present in many countries, Mexico, Americas, in Europe, in Jordan and Israel. Actually, Van der Vossenberg from the NVWA in the Netherlands, they started with a next strain project in which they sequenced TOBRV isolates so that you can see whether isolates are related or not, whether they are related or not. And there is a big blob on the Netherlands because it's a Dutch group, and so the Dutch isolates are overrepresented, but also there are a few isolates from the Middle East. We know already from our diagnostic labs and from seed lots that also in South America and Middle America there was already for quite a long time a TOBRV. So it's more logical that in the South and Middle America there might be more sources of sequences, but those are not yet into the equation. So there is genetic diversity, and well, that is quite. It's the same with COVID. Of course, there you have many more sequences, and so there is a better knowledge of the genetic diversity for COVID. But both viruses are highly variable, and there is debate about the origin. COVID, I think Leos was discussed. And for TOBRV, I think it's more logical that the Middle and South America are the origin. So what about pathways of TOBRV? Clearly, seeds is a pathway, and we have found highly contaminated seed lots over long distance spread. Seed is a pathway, but there are other sources we know by now, and also fruits from different countries were imported, tested, and there were high yields of TOBRV on fruits. So those apparently reach the market and can be a source of introduction as well. Smoking, of course, people spread the virus as well. So there are quite a lot of different pathways of the rapid TOBRV spread. Well, in conclusion, Tobamal virus are very persistent. That's maybe a difference with COVID, and very abundant. They come by trillions, and I think that's our problem. Qualitative validation have been done with natural contaminated seed, not yet in copies per ml, such as in COVID. So I think we can do better there and learn from medical world. Molecular TOBRV specific tests have been implemented for seeds and leaves, and we pick up seed lots. And well, we know now that there are alternative pathways as well. The sampling is critical for TOBRV and COVID. The interpretation of TOBRV data, it's not so easy what to do with the values of the relatively high values. Is that still biologically relevant? And we know the virus is so persistent, and disinfection of processing equipment is difficult. So it's hard to do a good interpretation of those data. And what we see is that TOBRV spreads rapidly and it evolves rapidly, and there is an increasing prevalence. Well, maybe the very good news is that there are the first resistant varieties are on the market, so breeders are working hard, and eventually probably this we will cope with this new challenge. Well, that was all. Rijkswaan did some good work with the BVS2 single-strand DNA, and NVWA did quite a lot of work on sequencing and also on the sampling experience with the tissue-specific expression of TOBV in the different tomato parties. That's all.
Thank you so much, Harrie. This was very interesting. You told us the stories of the TOBRV, with all the diversity, quantity on the leaves that is incredibly high, but is very interesting. So thank you so much for this. I'm afraid we don't have a huge amount of time for questions, but I'll try to go through a few questions. I believe there is some of the questions has already been answered by the speakers. Dr. Loh, thank you so much for having answered, and Dr. Ute as well. There is one last one that just came now from Roland, and it's to you, Harrie. Would increased quantification of the virus the relevance of detection to a degree that the high CTs can be disregarded, maybe in comparison with transmission studies or bioassays?
Yeah, personally, I think that the relatively high CT values, that those virus lots are, or those seed lots are not, that the chance that there will be seed transmission is low. But yeah, you need to check that with with grow outs, and that's quite laborious. But I think probably we overestimate the risk with those seed lots. But yeah, it's still in respect of if a pathogen is still not there, I think you want to avoid any risk. So the perspective of MPPOs and other for the seed industry, maybe it's not completely the same.
Thank you so much, Harrie. So I would like to have a question to perhaps Dr. Ute or Irena can answer. Is if a product is changed if it was pre-qualified, does the manufacturer have to simply notify such a change or also submit the respective validation data?
It's basically very much depending on the change notification. However, if it's affecting the design of the product or the intended use claim in regard to specimens or population to be tested, we require supporting data to support such a change notification. And I have put the link in the chat for our website where we have more information about reportable changes, and they apply to pre-qualified as well as to emergency use listed products.
That's great. Thank you so much, Ute. Dr. Loh, how can objective performance tolerance for a qualitative test that incorporates clinical performance be determined?
Sorry, can you repeat the question?
How can objective performance tolerance for a qualitative test that incorporates clinical performance be determined?
Okay, so what we shared earlier was an approach whereby we first define the desired predictive values, the positive and negative predictive values, and then we would then apply those predictive values on the ROC curves that are constructed when we validate the assay. So oftentimes when we validate the assays, we would have a pool of samples that we know have the disease, and then we would have a pool of samples that we know are healthy controls. And then we would run these two pools of samples through the assays, and then we would determine the numerical values and then interpret it as either positive or negative. The interpretation of whether it's positive or negative can be refined or informed by the construction of the ROC curve. So once we have constructed the ROC curve and when we apply the positive or negative predictive values, we will then form the boundary of the performance limits, which we can then use to monitor the assays. If I can call up my screen and if I can just move down. So this is what we often get when we first do so these are a group of known healthy samples and known positive samples. And when we measure them, we often get differing numbers, either CT values or, for example, COI values on the the immunoglobulin assays. And then we could then use this and vary the threshold here to determine what is the false positive and false negative rate, right? And then we would then be able to construct this ROC curve. And then the manufacturer recommended cutoff or the laboratory chosen cutoff is then plotted on the ROC curve. Then once we have put in the appropriate positive and negative predictive values, this would become the boundary of the performance limit. And then when we have a positive bias, it would actually move the cutoff or the threshold in this direction. And if we have a negative bias, it will move the threshold this direction. And once the threshold is moved beyond the chosen predictive values, then it is considered as no longer fulfilling the desired clinical performance. Does that address the question?
Yes, thank you so much, Dr. Loh. I received a question here, Harrie, for you. This is a quick one. Perhaps you can answer maybe in a minute. How the qPCR positive results were confirmed? Actually,
you can use a sequence analysis to confirm a result. The problem is if when the CQ values are relatively high, then the sequence analysis, the additional sequence doesn't work. So that's only possible for seed lots with relatively considerable virus load. You can do additional sequence analysis. There's also one additional TACM PCR that is in use by several laboratories. That's the Winter and Menzel TACM, which is actually a third TACM that can be used to confirm the presence of TOBV.
Oh, that's very good. Thank you so much. I'm afraid that we just run out of time. It has been such a wonderful event, and I really appreciate all the speakers, of course, all for the excellent presentations, and for the audience for being here, for the questions. And I would like to say a special thank you to a member of my team, Dr. Chantra, for the scientific organization of this event. I really hope that everybody has learned and enjoyed as much as I did. And I think it was fantastic, incredible to have all this different experience putting together, and of course, enriching the work that we do inside of the ISHI community, inside the SF. And I really hope that we can take this going forward. And of course, we're going to have much more opportunity to cross-pollinate it with some other colleagues on different fields, so we can learning together and of course, enhancing the knowledge that we have. And with that, I would like to say thank you to all of you, and stay safe and stay healthy. And let's catch up in the next opportunity. Take care, everybody.